Hello everyone, I'm Gayatri from 2nd MSc Microbiology, Sacred College, Tirupattur. Today we are going to discuss about microbial assay of antibiotics. Let me see about a small definition for microbial assay of antibiotics. A microbial assay of antibiotics is a bioassay technique used to measure the potency or concentration of an antibiotic by observing its effects on the microorganism. This method involves exposing the standardized strains of bacteria to various concentrations of antibiotics and then measuring the growth inhibition. Used to determine the potency of antibiotics, compare the growth inhibition of test organisms by standard antibiotic with that of unknown sample. The results are typically compared to the standardized reference to determine the antibiotic's effectiveness. This technique is crucially in enduring the quantity and efficiency of the antibiotic medication. The principles of antibiotics in microbial assay are dose-response relationship, which means the incubation of microbial growth is directly proportional to the concentration of antibiotics. Then, standard curve, a graphical representation relationship between antibiotic concentration and microbial growth incubation. Comparison. The potency of the unknown sample is determined by comparing its effective on microbial growth to standard curve. There are two methods of microbial assay of antibiotics. One, cylindrical plate or cup plate method. The principle of cylindrical plate method is that an antibiotic diffuses from the cylinder or a well into an agar medium inoculated with a standardized bacterial strain, creating zones of incubation proportional to the antibiotic concentration of potency. Procedure for couplet method. Prepare a nutrient agar and inoculate it with the test organisms. Pour the inoculated agar plate into a petri dish plate. Place a sterile cylinder or cups on the solidified agar. Add unknown concentration of standard antibiotics and the test sample to the respective cylinders. Then, incubate the plates under appropriate condition. Measure the diameter of the incubation zone around each cylinder. Plot a standard curve using the logarithm of antibiotic concentration against the diameter of incubation zones. Determine the concentration of test sample from the standard curve. The second method is turbidimetric method or a tube method. The principle, the growth of the test organism in a liquid media is measured by turbidity or addition of antibiotics inhibits the growth and leading to decreased turbidity. So procedure, prepare a series tubes containing the different concentration of standard and test antibiotic solution. Inoculate the tubes with the test organisms. Incubate the test under appropriate condition. Measure the turbidity of the culture using spectrophotometer. Plot a standard curve using the logarithm of antibiotic concentration against the abundance or optical density. Determine the concentration of test sample from the standard curve. Factors: the test organism should be sensitive to the antibiotics, grow rapidly and reproducible. Media composition supports the growth of test organisms and allow the optimal antibiotic diffusion. Incubation conditions, temperature, humidity and incubation time should be controlled. Antibiotic stability. The antibiotic should be stable under the assay conditions. Application: quality control of antibiotic products, standardization of antibiotic potency, determination of antibiotic susceptibility of microorganisms, research and development of new antibiotics. Precautions are: adapt to the aseptic techniques to prevent contamination, use accurate measurements devices for antibiotic concentration and volumes, ensure the linearity and reproducibility of the standard curve, employ the statistical methods for data analysis. Data collection and analysis. Calculate the mean diameter of incubation zones for each antibiotic concentration. Plot a standard curve using the logarithm of antibiotic concentration against the mean diameter of incubation zones. Determine the concentration of the test sample by interplotting its incubation zone diameter in standard curve. Express the potency of the test sample in appropriate units. Examples: IU by mg, mu g by ml. Quality control. Use validated standard antibiotics. Participate in interlaboratory comparison studies. Monitor assay of performance through control charts. Thank you so much. Thank you so much for your patience learning.